Hello, plant friends. Welcome. I'm so excited about this lecture. Actually, this is a two-part lecture because there is so much information that we're going to cover. I'm going to break it up into two different segments. So welcome to segment 1 of recognizing the sexual reproduction of plants. We're talking about seeds here. Seeds are so magical. It's one of the most fascinating parts of plant growth and development to me. What a seed contains, as we will see together, is everything that it needs to start a new plant. So as we move forward, please take the time to write down your questions and then post them to our Blackboard discussion when you're done with this lecture. Let's get started. Let's begin by thinking about the seeds that you yourself have seen and interacted with. Now, assuming that you've never planted a seed before, think about the seeds that you've dealt with in your food. So, for example, pepper seeds that you have maybe removed from the inside of a hot pepper to not have so much heat in your food. We know what those little seeds look like. They're kind of flat, sort of a squashed. It would be kidney shaped if it were longer, but it's a little bit more round than that. Now let's think of that versus a cucumber seed or a pumpkin seed or a watermelon seed. Now let's think about those versus a banana seed. So those little dark dots inside the banana versus a strawberry seed. Teeny, teeny, tiny little things. And then we look at a much larger seed, for example, a sunflower seed. All of these seeds, shapes and sizes are going to correspond with the family of plants that they come from. So we know that there's a huge variety of sizes, shapes, and colors. We know that there's some that are teeny, teeny, tiny, like our strawberry or our basil seeds, and then others are much larger. Like I said, our sunflower seeds, pumpkin seeds, they have different shapes, they have different sizes, they have different looks, and they also come in different colors. Regardless of the shape and size, seeds have one purpose, and that is to grow a new plant. So as I mentioned a moment ago, the inside of every seed has all of the resources it needs to generate a new life. Now, what it needs is favorable conditions for the seed to germinate. Now, real quick, germination is the sprouting of a seed. So when we are planting seeds, we are trying to promote germination. Now, each different type of plant is going to require different environmental circumstances in order to germinate. But very basically, an ovary needs to be fertilized in order to create a seed, and that happens through the process of pollination. So when the male genetics and the female genetics are combined, they result in the formation and development of seeds. And so that seed is going to contain the genetic information from both plants. So when we talk about sexual plant reproduction, we are talking about using seeds to reproduce more plants. So in this lecture, we are going to talk about the different plant parts. We're going to talk about the processes. We're going to talk about formation and development of seeds, as well as the advantages and disadvantages of using seeds for plant reproduction. Then we are going to go into part 2 of this lecture and discuss cross-pollination. Beginning with the anatomy of a flower, if a flower contains the following 4 parts, it is considered to be a complete flower. So number 1 is sepals. These are, we've talked about this before. So these are sort of leaf-like structures that are actually there to protect the bud when it is closed. So once the flower opens, those little green appendages basically underneath the bottom of the flower are the sepals. Collectively, the sepals are referred to as the calyx. Now, obviously, we know the petals. These are going to be the bright, beautiful, showy part of the flower that have color. They've got scent to them at times, and both of those features are meant to attract pollinators. Then we have the stamen. Those are the male parts of the flower, and the pistils, the female parts of the flower. Here we take a look at a graphic representation of those parts of the flower. Please take a moment to pause this video and sketch a flower labeling all of the different parts. And we see here that it has the sepals, it has the petals, it has the pistil and the stamens, and that means that this is a representation of a complete flower. The stamen is the male reproductive organ of a flower. If we were to draw a comparison between plant bodies and animal bodies, the pollen that is held on the stamen is essentially like sperm. So this is what is going to fertilize an ovary to create a seed. The stamen itself has 2 parts, the filament, and that's the stalk, if you will, and the anther, which is what holds the actual grains of pollen. The pollen itself is usually a yellow to a orange or brown rust color, and each individual grain of pollen has the complete genetic material of the parent plant. The pistil is the female part of the flower. It's sort of vase-shaped. It's in the very center of the flower, and it is made up of the stigma, the style, and the ovaries. Inside the ovary is individual ovules. Each ovule, if fertilized, has the potential to create a seed. Starting at the top of the pistil, we have the stigma. So this is actually physically sticky so that it can catch and hold pollen. If we look at the image on the right, this is a perfect representation of a stigma. We can see the little yellow orange-ish spots. Those are grains of pollen that have actually been stuck. Now, if you have any flowers around your house or your yard that you can go and look at, if it has a large pistil and it has a fairly big stigma at the top of it, if you very lightly touch it with your finger, it does have a little bit of, if not stickiness, wetness, because we always come back to the fact that a plant wants to be as advantageous as possible in its reproduction. So it will create different features and characteristics in its reproductive organs to make the most possible seeds as quickly and efficiently as it can. So part of that is having a sticky stigma that catches and holds individual grains of pollen. Then when we move down, we have the style, and that is actually a tube that the pollen grains will go down. It will create a pollen tube until it reaches the ovary. Once the pollen grains make their way down through the style, they reach the ovary. The ovary has the ovules or the egg cells, and upon fertilization, they start to create seeds. So if you look closely at this cross section that we have here on this slide, you can see that each individual seed or developing seed has basically, and again, I like to sort of compare these plant features to human and animal features. Basically, what we see is the equivalent of an umbilical cord, but that is the pollen tube that has made it down from the stigma through the style and into the ovary to attach to the developing seed. Quick note about terminology. So with ovaries, there are superior and inferior ovaries. A superior ovary is going to be above the calyx of sepals. An inferior ovary is going to be below them. And it's important to be able to identify each one of those because that's going to help you with your plant identification down the road, right? So again, if it is above the calyx of sepals, it's a superior ovary. If it's below, it's an inferior ovary. So pop quiz, rewind and go back to the last slide with that picture with the cross section. Is that a superior or inferior ovary? We already know that a complete flower has all four of the floral parts: the petals, the sepals, the pistil, and the stamen. So we can derive from that that an incomplete flower would be lacking one or more of those floral parts. So that's complete versus incomplete. Now perfect flowers versus imperfect flowers depend on, do they have one of the sexual reproductive organs, male or female, or do they have both? A perfect flower has both stamen and pistils. Now that's regardless of whether or not they have sepals or petals. So that means we can have an imperfect, incomplete flower versus a perfect, complete flower. Here is a beautiful representation of a female flower on the left and a male flower on the right. So we see that very distinctive shape of the pistil in the female flower. And then we see on the right-hand side in the male flower, a perfect representation of that stamen. So we have the filament that has the pollen-filled anther at the end of it. While most plants produce perfect flowers, there are, as we've seen, many examples of imperfect flowers that have either male or female reproductive organs. Now, it's important to note that the same plant might have imperfect flowers of both male and female. Perfect example of this is squash and zucchini. As we've said, pollination and fertilization are necessary for the formation of seeds. Now, those reproductive organs are not mature and ready to make new seeds until the flower opens, which, as a side note, means that we can't pre-pollinate our flowers before they've actually opened up. we said, each pollen grain has the genetic characteristics of a plant, as does all of the ovules inside the plant's ovaries. Now, depending on the plant, each ovary may contain one or more ovule. So there are some ovaries that will only create one seed, while other ovaries will create many seeds. An example of this is a tomato. We know that there are many seeds inside that tomato. Just like the pollen grain, the ovule contains all of the genetics of that parent plant, and the flower and the reproductive organs have reached maturity when the stigma becomes sticky. Pollination itself begins when a pollen grain comes into contact with the moist, sticky surface of the stigma. This can happen through mechanisms of gravity, insects, wind, or animals. So, if a plant self-pollinates, it means that the pollen comes into contact with the stigma in the same flower or other flowers of the same plant. So that means that we have the same genetics going into that pollination. Whereas cross-pollination is when we have pollen from one plant pollinating the flower of another plant. So there we have a hybrid. We have a cross of genetics. As we discussed in a previous lecture, when we have seeds that come from the cross-pollination of two separate plants, we have a blending of that genetic material. So there are going to be characteristics present from each parent plant, and the offspring is not going to be identical to either. We know that pollination occurs when a grain of pollen lands on the sticky surface of the stigma, and then it grows a pollen tube down the style. Now, fertilization happens when the male sex cell in the pollen tube comes into contact with and unites with the female sex cell in the ovule. So after this fertilization happens, those two sex cells will combine to form an embryo within the ovule. As development continues, the ovary will develop into a fruit or the seed pod. Now, it's important to note that each fertilized ovule develops into its own seed. So let's take a moment, use some critical thinking, and consider how some flowers have the ability to produce many seeds versus some flowers having the ability to produce one seed. So for the plants that produce one seed per fertilized flower, it means that there is a single ovule in that flower's ovary. Now let's think about other types of flowers who are able to produce many seeds within its ovary. So we think about tomatoes, peppers, cucumbers, things of that nature where one fruit comes from one flower, but each fruit contains many seeds inside of it. Now what that means is each one of those seeds, which was originally an ovule, and each one of those ovules was fertilized by a separate grain of pollen, right? And so then what we end up with is a fully formed, well-fertilized fruit with mature seeds inside of it. So every once in a while, especially with squash and zucchini, there will be a fruit that has an odd shape to it. It's almost like the end of it is I think of like an elf shoe almost. So what we end up with is a zucchini that's shaped like this, right? So typically, what causes that is it's been underpollinated. Some of the ovules have been fertilized, but some have not. And that would be a result of low pollinator traffic, right? So it's just kind of fun to think about the possibilities of having, multiple fertilized ovules in just one fruit. And again, if we think about a pepper plant or a tomato plant, where throughout its life it produces hundreds maybe of fruit, and so each one of those fruit contains more than a dozen at the very least seeds inside it. So it just kind of blows my mind to think about how many individual ovules are produced by each individual plant throughout its lifetime. And that always brings it back around to, what is a plant trying to accomplish? We'll talk more about that in a bit. Now, when we look at the anatomy of a seed, as we said before, each seed has everything it needs inside of it to produce a new plant. So inside is a tiny plant, and it has a food supply in there to get the plant through its first period of its life before it's able to start photosynthesizing and pulling nutrients and water through its roots on its own. So the first thing that we look at is the outside. We have that protective layer, a seed coat. Now, depending on the plant, seed coats have different thicknesses and hardinesses, and some are able to germinate with very, very little influence, and then other seed coats require a lot more work to break it down and allow the seed inside it to germinate. Then inside the seed coat, we have the three basic parts. We have an embryo, that's the baby plant. We have the endosperm, and that is the food source. And then, of course, on the outside, our seed coat. So that endosperm, the baby plant, it consists of leaves, a stem, and roots. Now, those leaves are referred to as cotyledons. And when we get into botany, we'll talk about this at great length, but it either has one cotyledon, and that is a monocot, or it has two cotyledons, and that type of plant is referred to as a dicot. And then it has its first root already in there. So during germination, these parts start to form inside the seed, and when the seed comes to maturity, that endosperm is ready to go, and the seed coat is constructed in such a way that it's going to protect the insides for as long a period as possible. So let's think about, as we've mentioned, wildflowers. Those wildflower seeds sit on the surface of the soil after the parent plants have died, and then they don't actually germinate and start to create new plants for several months. So those seed coats are very important to be able to preserve the inside parts of the seed until the environment is appropriate for it to germinate and start to grow. The majority of the contents of that seed is the endosperm. So like I said, this is where the nutrients come from. Mostly starch, there's some oil in there, some simple proteins. So these are the nutrients that, like I said, are going to supply the germinating plant while it is starting to grow until it can photosynthesize for itself and its root system starts to develop to the point where it can bring in its own nutrients and water supply. Now, if we think about an example of an endosperm with the starch and the oil and the protein, a perfect example of that is our pumpkin seeds that we like to roast. Also, sunflower seeds, perfect example of the majority of that seed being a food source. So that protective coat on the outside of the seed might consist of one or more layers. Those layers may be thin and easily broken, or they might be thick and hard. Different plants and their different seeds that have different kinds of seed coats might require extra steps in order to get them to germinate. And so a word that I want you to think about now that So what does a seed need in order to germinate? Put very simply, there are certain environmental conditions that need to be met for each particular seed. And these environmental conditions are going to depend on moisture availability, gas exchange, temperature, and light. And there are some seeds that require a period of dormancy. And as I mentioned with scarification, some seeds will need to be exposed to certain environmental effects or actions. An example being soaked in water or passing through the digestive system of another animal. Some seeds require being exposed to fire or smoke. We'll go into a little bit more on this. Now, this is where I like to pause for the cause and talk about plant psychology and evolution. So, for the purposes of this class, we agree that we all believe in evolution, right? So, let's talk about how plants are able to make decisions. Plants don't have brains like animals that will say, it's time to eat, it's time to sleep, it's time to mate. Instead, what plants have are eukaryotic cells. A prokaryotic cell, like most of the cells in our body, don't have a nucleus. A eukaryotic cell has a nucleus. So what that means is every cell in a plant's body essentially has a brain. And the plant makes decisions for what happens next in its life cycle based on the hormones that it starts to release and exchange throughout the tissues of its body. So we've already talked about auxin and the growth hormone, and we know that auxin helps the plant to make decisions about what tissues are going to be formed with the next cells. Is the next cell going to create a root? Is it going to create a leaf? Is it going to create a reproductive structure? Is it going to go into the process of scarring over, healing a wound that's happened on the plant, right? So hormones are messages that happen inside of the body of the plant. Same thing goes for us. Our hormones make decisions for what is going to happen in our bodies. So the purpose of a plant, let's think about this. What is the one purpose in a plant's life? To create more plants, to further the species, to continue its genetic material, right? So plants have evolved to use the most efficient and the most guaranteed method of propagating itself, right? So let's think about a couple of different examples. Let's start with succulents. Many succulents propagate best asexually. So they create a little plantlet, they separate off of itself or drop little baby plants. So we've got, for example, the mother of thousands, all of those tiny plantlets that are growing adventitiously, just waiting to drop on something where it can take root and grow, right? And then we've got our agave and our aloe plants that grow little pups around it. Why would succulents focus on asexual propagation? Maybe because in an arid climate, there's not a guarantee that pollinators are going to be present, right? So if there's not a guarantee of birds and bees to be there to help it fertilize its flowers and create a viable seed, side note, most succulents do flower at some point, and the flowers are gorgeous. But again, it's not guaranteed that they're going to have something to fertilize the seeds inside of the flower, right? So those plants have evolved to propagate as often and as vigorously as possible. It's got to play the numbers, right? Now let's look at fruit, sexually propagated plants. Let's think about a papaya, right? So the papaya is pretty big. It's got a lot of water in it. It's got a lot of sugar in it, and it's got fairly large seeds. Now that plant has evolved to be attractive to mammals. Why? Because that is a larger plant that is not going to do as well for its community of papaya plants if all of the fruit just drops directly below the tree and then grows right there. They're going to be too close together. They're going to choke each other out. And the plant knows this in its cells. So what it wants is for its seeds to be carried away and deposited elsewhere. This is where it gets really fascinating. Mammals like to get a buzz. Maybe we know this from experience. Maybe we know this from seeing if we left our jack-o'-lanterns out too long in the heat of a Texas fall and they start to ferment a little bit, and then all of a sudden our property is full of drunk squirrels. Mammals like to get a buzz. So when sugar ferments, it creates alcohol, and it also creates a smell. We can smell that fermentation. And so mammals smell that and they immediately want to go there. Now, they're not going to bother picking out the seeds of their fermented fruit and pumpkins, right? They're going to eat it all and then they're going to carry it away, right? After they carry it away, they're going to pass those seeds in a perfect planting medium. We know what I'm talking about. So if we have this papaya in the jungle and a primate comes and eats it and then carries it away, it's got a little bit of a buzz, right? And it got water, it got fiber, it got sugar, it got nutrients, and it ate a bunch of seeds. It's going to deposit those seeds far away from the parent plant, which is the objective. That's what it wants. It's going to deposit those seeds in a nutritious and dark and warm substrate, we'll say, in the poop. So what the plant has accomplished is being carried away and planted in the correct medium in a place that's far away from itself, right? So this is where we get into why plants have evolved with different sizes of seeds that need different planting circumstances. And we'll get into this in our seeding lab for sure. But as a real quick preview, larger seeds usually come from larger fruits, right? Which means that it is going to be eaten by a mammal, sometimes a larger mammal, and that means it's going to be passed and deposited in a larger pile of substrate. So that seed has evolved to need darkness, right? To need darkness and a consistent temperature because it's insulated in that matter. Once it has those circumstances, it's going to germinate versus, we'll say, like a little tiny seed. So let's talk about actually brassicas. So this is the cabbage family, right? So if you've ever seen flowers that come off of cabbage or kale or broccoli, things like that, they're very light, they're very papery because they typically don't get eaten by anything. They get carried by the wind. And so they have evolved to have basically little propellers on them where when the wind catches it, it carries it away and disperses those seeds a little bit further. So those seeds are tiny, they're light, and they are intended to germinate after they've just fallen on the ground. So those seeds, we don't want to plant very deep because they have evolved to recognize that they need sort of a light and dark period, a day to night period, and that's going to trigger their hormones to start to germinate. And they also need a little bit of variation in temperature. So those big seeds, they want insulation and no variation in temperature, whereas the small seeds, they actually need variations in light, dark, and then high and low temperatures in order to germinate. Let's move on. In discussing asexual propagation, we definitely talked about there are some plants that propagate better from cuttings or divisions. They have more success propagating asexually. Now, there are other plants that propagate best from seeds. Many of these plants are going to be our annuals. So they go through an entire life cycle in 1 year. They go from seed to seed in one season. So examples of these are going to be our typical agricultural products, our tomatoes, our peppers, our squash, zucchini, etc., etc. Now, the quick caveat there is, as we'll talk about in botany, annuals are sometimes defined regionally. So it is possible to perennialize a tomato plant or a pepper plant in the right environment. Whereas in Central Texas, typically when we have them planted outside, they do only last for 1 year. Let's take a few slides to discuss advantages and disadvantages of seed reproduction. So at the top, starting with advantages, using seeds will allow a grower to produce a large number of plants. Let's remember that tomato plant. Each individual tomato has possibly dozens of seeds. If those seeds are mature and viable, that means from each individual tomato fruit, we would be able to produce dozens of individual tomato plants. And that, for those types of plants, is going to work out a lot better than taking dozens of cuttings to try to propagate tomato plants that way. So as I said, annuals reproduce quickly from seed because that's what they've evolved to do. And there are some plants that only reproduce by seeds. If you have ever grown a squash or a zucchini plant, you might know that none of those tissues would be good for asexual propagation. So really the only way to do it is from seeds. This is also a cost-effective method of propagation. For the most part, and there's always exceptions, seeds are somewhat inexpensive and propagating from seeds requires less labor. So it also uses production space more efficiently than asexual propagation because those seeds are typically fairly small. They can be grown close together in a seed tray without interfering with the cells next to them. So we would be able to get a higher number of individual plants in a smaller amount of surface area. If stored properly, seeds can remain viable for a fairly long period of time, which is really great because often if you buy a package of seeds, you're buying up to hundreds of seeds in one small package. And even if you don't want to grow, we'll say, hundreds of lettuce or basil plants just for your own use, you don't want to throw all of those seeds away if you don't have to. So under proper storage, you would be able to return to that bank of seeds and only plant what's needed. I hear you asking, but beans, what is proper seed storage? It needs to be cold and it needs to be sealed away from moisture. So let's remember that one of the environmental circumstances that will cause germination is presence of moisture, right? So what I always recommend is that you get your seeds, and whether they're in packets or Ziplocs or whatever, you put that in a paper bag, and then you put that paper bag in a sealed glass jar or a glass Tupperware that snaps. Interestingly, and I didn't find this out until, I would say somewhat recently, but it's been in the last few years that plastic bags and plastic Tupperware, they're not actually moisture tight. If you have, a Tupperware and you put a bunch of pasta sauce in it and you turn it over, it's not going to spill out, we hope, necessarily, but over time, moisture will get in and out. So if we're thinking about saving seeds from 1 year to the next, we want to make sure that it's as moisture tight as possible. And we also know about refrigeration and how that is an interaction of moisture in the air. So if we think about things in our freezer that can get freezer burned, or when frost builds up, the same thing will happen on the inside of your container that your seeds are in. So My favorite method is to just get a glass mason jar and then seal the top really tightly. And then that really should work for at least a year, if not multiple years. And as always, it depends on the plant. Back to the slide. As we discussed in a previous lecture, reproducing plants from seeds allows for the potential of crossing genetic characteristics for a more vigorous plant. We call those hybrids that have the characteristics that we really want out of the new plant. Another advantage, and this is very significant, is seeds are typically disease-free. If a parent plant has a disease, and those are going to be fungal, bacterial, or viral diseases, typically, especially for fungal and bacterial infections, those are not passed on to the seed themselves. Viral infections are a little bit different because they actually get into the cells of the plant and can start to affect genetic makeup. And of course, there are some disadvantages for reproducing plants from seeds. Number one, and this ties into the advantage of using asexual propagation, is some plants do take a long time to grow from a seedling to a saleable plant. So that means there's a higher production cost for the grower. The longer you have a single plant or a single tray of plants taking up space, nutrients, water, and time in your greenhouse, the more money it's costing you because you aren't able to put any new plants right there in their spot. Next, we don't know exactly what characteristics will be coming from our seeds, especially if they are hybrids that we have created ourselves. So a piece of advice that I always give when talking about seeds and propagated material is know where it comes from. I know that there are some really, really wonderful individuals who sell seeds and plants on Etsy, but you don't know what they've been through. You don't know how they were procured. You don't know what genetic material is in them. You don't know how they were handled, if they are clean and disease-free. You don't know what it's gone through, so you don't have a guarantee on what you're going to get out of it. And in this class, we talk somewhat specifically about what it takes to grow in a greenhouse for profit. So if we are trying to make a profit and get uniformity in the plants that we're going to market and sell, it's really important that we do know about what that plant is and what it's been through. And now lastly, one disadvantage for using seeds is some of them require very special handling and treatment. So if it's a teeny tiny seed, for example, begonias, begonia seeds are basically like dust. And if you look at them too hard, they will disintegrate in your hand. Very, very difficult to handle. And then, like I said, scarification treatments might have to be performed, as well as another term, stratification. So those will help speed the germination process. But again, we have to think about each seed that has to be handled individually and put through a special process is going to take extra time. Your time is your money. So the longer we spend handling seeds, the more money we have put into each individual plant. That covers part 1 of our sexual propagation lecture. Thank you for your time. If you have any questions, please go ahead and post them to the discussion board and then get ready for part 2. Thanks.